Hello, everyone. My name is Flora, and after learning research secrets at UCSF and UC Berkeley, I started a group at Stanford University. And we are interested in understanding the origins of cell signaling mechanisms that enabled the evolution and origin of animals. And in particular, we're going to be focusing on tyrosine kinase signaling to start our research projects off. Because tyrosine kinase signaling is a fundamental cell signaling pathway in animals, what has recently been appreciated due to work by pioneering biologists such as Nicole King and others that study the unicellular relatives of animals, tyrosine kinase signaling actually is predicted to be present in the unicellular relatives of animals, which means that the ancestor of animals very likely had the capacity to perform tyrosine kinase signaling in a way that we now see is very fundamental to the biology of animals, and when it's misregulated in animals leads to disease. And so we are using choanoflagellates, and this is also a model that's used by another this generation faculty, David Booth, who is unable to be with us today, to study the ancestry of tyrosine kinase signaling. We are really excited to approach this pathway because we know that receptor tyrosine kinases are predicted to be present in these organisms, and they also are associated with additional protein domains that are known to regulate cell signaling. What we are also very much excited about due to things that we found doing immunofluorescence staining is that tyrosine phosphorylation also seems to be a component of development in choanoflagellate biology. However, we don't know how this is regulated. We just know that this happens when we form colonies in various colonial choanoflagellate species. But we know that they're likely to be regulated because we also have the presence of adapter proteins that we know regulate this signaling pathway in animals, also present in choanoflagellates. We're also really excited about the biology that intersects bacteria and eukaryotes, and in particular looking at bacterial interactions that play fundamental roles in the development of choanoflagellates. So this multicellular development that we see in choanoflagellates is actually induced by bacterial signals, and work by Nicole King and others have identified what those bacterial signals are, but we don't know what the choanoflagellate receptors are for those ligands. And so we're really interested in looking at this as an aspect of biology because we know that animals originated in this diverse environment with bacteria playing a very fundamental role. And we know that we see hallmarks of that today in things like the microbiome. And so we think that there's a lot of biology still to be uncovered in this area. And so, as I mentioned, we just launched in September, and we're looking forward to pursuing this area, and we're also actively recruiting. So grad students and postdoc, please talk to me, and I'm also interested in research collaborations with others that are doing great work here. Thank you.